this afternoon. It's my great pleasure to introduce Chris Brackley. He's from Edinburgh, and he's going to tell us all about clustering of DNA and chromosome binding proteins.
Thank you. So I'll just start by thanking the organisers of the conference for giving me this opportunity to talk today, and I should acknowledge the other people who've been involved in this work, who are Davide Marandozzo, who I work with in Edinburgh, and also Peter Cook from Oxford. Okay, so I'm going to talk about something maybe a little bit different from what we've been hearing about. So I'm talking about clustering of proteins which bind non-specifically to DNA, and we've been using coarse-grained Brownian dynamics simulations to look at that. And my talk's going to be in two parts. So I'm going to start by showing you some results from some very simple models of proteins which cluster. And then in the second part, we've been doing some larger simulations where we're trying to compare what we see there with some experimental results. And so I'm going to start just by, as way of motivation, just reminding you of some situations where you see clustering of DNA binding proteins. And so this is in things which have already been mentioned already this week. So as we all know, the DNA inside a cell is highly confined. So in bacteria, you have millimeters of DNA in a cell size of microns. And in eukaryotes, you have meters of DNA inside cells of tens of microns. So the DNA has to be confined. And in bacteria, here are just some examples. The DNA forms loops, often due to the action of proteins, which can also act as molecular ties. So you have the nucleoid-associated proteins, or NAPs, which we've already heard about, which could facilitate the organization of the bacterial chromosome. Transcription factors are also themselves thought to be involved in creating loops in the DNA. And then there's proteins such as the histone-like nucleoid structuring protein, which has been seen to dimerize and form bridges between different DNA segments, and they're often found in clusters in the center of the nucleoid. And also there's growing evidence that transcription occurs in localized regions or factories, and such transcription factories would basically be clusters of proteins, which are the machinery involved in transcription. Okay, so in eukaryotes, again, we've heard about this earlier in the week. So the DNA is formed into chromatin by winding around histone proteins to form nucleosomes. And so you get a 10 nanometer fiber of basically a string of nucleosomes, and as we've already heard, that forms into a 13 nanometer fiber, and then these chromatin fibers are also themselves compacted further, and this is possibly also due to protein ties. And for example, during interphase, each chromosome has its own territory with a non-random position within the cell, and the nature of further organization within those territories and also between the chromosomes is still a focus of a lot of research. And again, there's some evidence that transcription occurs in factories. So for example, this picture, these colored in green here is newly transcribed RNA. So the fact that these RNAs are found in foci suggests that they were produced in concentrated regions or factories. Okay, so this is just some examples of where proteins are found to cluster. So I'll briefly run through our coarse-grained simulation model. So we've got a very coarse-grained model, but it's quite standard. So we treat the double-stranded DNA as just a chain of spheres, so treat it like a semi-flexible polymer. And each sphere or bead has a diameter of about 2.5 nanometers. So each of these monomers represents about 8 base pairs of DNA. Okay, and then we do Brownian dynamic simulations. So we just numerically solve a Langevin equation for each of our DNA beads. And this term with this V in it here gives a force as a result of the potential experienced by each bead due to all of the other beads. And then there's a noise term, so eta here is a Gaussian noise with a zero mean that has no correlations. And so then this friction constant gamma gives our implicit fluid and determines the diffusion constant of the monomers. Okay, so now the potentials. So the monomers interact with just a steric repulsion, so as hard spheres. So we just used a simple truncated and shifted Leonard-Jones potential for that. The adjacent monomers are connected by strings, so finite elastic non-extensible springs. So that just means they can't move further apart than some distance r0. And then every two neighbouring links experiences a bending stiffness. So we use a Crackley-Porod potential, and we choose parameters so that there's a persistence length of about 50 nanometers. So this is right for naked DNA. Okay, so it's a sort of standard model of DNA as a simple polymer. And then we wanted to very simply model proteins. So we started with just the simplest model of a protein as just a sphere, so just an isotropic sphere. And we wanted that to have an interaction with the DNA. So this could represent, for example, just a Coulombic interaction with the DNA backbone. So we used just a shifted and truncated Leonard-Jones potential. So it looks something like this. So it's truncated at some distance r0. For that, we just chose a distance about the same as the Debye length for a Coulombic interaction in a cell. Okay, and then we can choose the interaction strength between the protein and the DNA. So the proteins just like to stick to the DNA. But importantly, we didn't include any attraction between the proteins, so we just had a Haar sphere repulsion between different proteins. Okay, so then we wanted to ask the question, what happens if we consider a large number of these proteins interacting with a long DNA molecule? So we started with these non-mutually interacting spherical proteins, and so the easiest way of describing what happened is just to show you a video. So this is a little video of a simulation. It's a 2D projection of a 3D system. So the blue here is this DNA molecule, and then the green are the proteins, which like to stick to the DNA, but there's no interaction between the proteins. Yes. So no, so the proteins here are shown a little bit bigger than they actually are. So they were about the same size as the DNA monomers in this case.
What is the ratio of the thermal energy?
Oh yeah, so the attraction interaction here is about 5 kT. Okay, so you can see that these proteins start to stick to the DNA, and then you can see them forming clusters. And if you look closely, as these clusters grow, you can see that they sort of form lines or rows of these proteins. So I've got the same video again, but this time, just to make it clearer, I've made the DNA invisible. So you can just see these proteins. They form these clusters, and then you can see more clearly that they're forming rows. So you see these rows forming. Okay, so why do the proteins cluster? So there's a couple of reasons. So because the spheres are isotropic and they can interact with the DNA all around, then the spheres can form bridges between two different segments of the DNA. And so by making multiple contacts with the DNA, they can reduce the energy by binding to the DNA. But there's an entropic cost of bringing two segments of the DNA together, and also the proteins can bend the DNA locally and cause deformations. So it turns out that it's better to put these proteins together so as to minimize the DNA distortion and this entropic cost of bringing together DNA segments. And also, once one protein has brought two pieces of DNA together, then that sort of leaves a nice, well, the DNA is just the right separation for another protein to sort of move into that gap. And I've got another little video here just showing this. So you see at the start there are two proteins which have brought these two bits of DNA together, and the thermal fluctuations are sort of pushing them together just because the DNA, different strands, want to move apart. And then there's a nice gap here.
But they never detach, so it's so deep the potential.
So when they're between two with this interaction, then they stay together. But sometimes if there's just one here, then the fluctuations are enough to kick it off. But we haven't really done enough simulations to find exactly what energy you need to put, what well depth you need to make them cluster. Okay, and we also measured the number of clusters as a function of time. So at the start of the simulation, we switched off the attractive interaction between the proteins and the DNA and allowed it to equilibrate. And then after you switch on the interaction, you see these clusters form. And if you look at the DNA protein binding energy, then as the proteins make more and more contacts with different parts of the DNA, then that energy decreases with time. Okay, so these rows are reminiscent of some structures seen with these HNS, the histone-like nucleoid structuring protein, which recent work has shown that these are thought to, because they dimerize, they can line up in rows, bringing two segments of DNA together. So our proteins were isotropic, so we wanted to try and do something a bit closer to this. So we looked at a slightly more realistic model for our protein where we had three spheres. So a larger sphere, the same size as before, which interacts only repulsively with by a hard sphere interaction with the DNA, and then two smaller spheres which are acting as binding sites, which are sticky for the DNA. And again, there's no attractive interaction between the proteins. Okay, and I think one more video. So here you can, it's less convincing than before, but if you watch, it takes a little longer. Then you can see them start to form clusters, and it's less convincing, but some of them are forming rows. So you can see the same thing even when we've decreased the part of the protein which can interact with the DNA. Okay, and then here's just an image from the end of that simulation, and the same one with the DNA made invisible, and you can see that there are some clusters that are sort of in rows. So it's less, so the clusters are smaller than before, and if you look at how the number of clusters changes with time, then for the bridgers, which is what we were calling these, there are more, so therefore smaller clusters, and it takes a bit longer for them to form. And if you look at the DNA protein binding energy, it takes longer for them to make more contacts with the DNA. Okay, so those were quite small proteins. So then we looked at larger proteins. So if you put in large spheres, which are so these are about 3 times the size of the DNA beads, then you see that the DNA actually wraps around the large spheres. So here you need a binding interaction of of around 10 kT to get them to wrap around. So this is sort of reminiscent of the nucleosome, where you have the DNA wrapping around the histone complex. Okay, and so we did a simulation putting lots of these large spheres in a simulation with a long DNA molecule, and no video this time, but we saw similar as before. So initially, when we first switch on the interaction between the spheres and the DNA, they start to cluster. And here, the clusters, they're more globular in shape compared to the rows which we had with the small proteins. And they're also a bit larger. Okay. And again, you can plot the number of clusters as a function of time, and also the binding energy, total DNA protein interaction energy as a function of time. So again, the clustering is happening because it costs energy to wind the DNA around these large spheres. So if you put several of them together, then maybe it can wind around more than one, still making contacts with the DNA, but not costing as much binding energy. There's another interaction in this case which could also be driving the clustering, and that's an entropic interaction, the depletion interaction, which is well known from solutions of polymers. So this basically, the idea is if you have a solution of large and small polymers, then there's some volume around the, colloids, that's what I mean to say. Large and small colloids, then there's some volume around the large colloids which the small particles can't enter into, so there's an excluded volume. And if two of the large particles touch, then there's an overlap between the excluded volumes, so this decreases the total excluded volume and increases the volume available to the small particles, so that increases their entropy. So there's an entropy-driven interaction between the large particles, and in our case, the DNA could be acting as the small particles, causing clusters or loops in the DNA to form by sticking together these larger particles. Okay, so again, our polymer was just a sphere, so it interacts isotropically. So we tried to make something that looks slightly more like a nucleosome core. So we built it up out of several spheres. So the green ones only interact with a steric hardcore repulsion with the DNA, and then the red ones are sticky for the DNA. So this is more of a cylinder shape, and the DNA sticks to the edges. And you see that again it wraps up, but if you put lots of those in the solution with a long DNA, then again you get these large clusters forming. Okay, so just making the DNA invisible again. So you see these large clusters of these histone-like cylindrical polymers. Okay, but as we know, chromatin isn't like this, so the DNA wraps around the histones pretty regularly, and you get these fibers forming. So why don't we see that? Okay, but similar clusters have been observed elsewhere. So for example, there's been some experiments where they put gold-coated nanoparticles in a solution with DNA, and the DNA wraps around the nanoparticles like it wraps around the nucleosome cores, and you see these clusters of nanoparticles with the DNA wrapped around. And so these globular clusters look a little bit more like what we see in our simulations. Also, there's been some experiments done some time ago where they try and they combine histone proteins with DNA in vitro, and if you use modified histone proteins, so I think in this experiments they so the histone proteins have these long tails which extend out from the nucleosome core, and these were modified to cut these tails off, and then you see these more globular-like clusters of histone DNA complex forming. So again, that looks similar to what we see in our simulations. Okay, so obviously our histone-like model proteins are too simple in order for them to form regular fibers. So you must need a more detailed description of them, and perhaps it's the tails which are important for that. Or an alternative way of modelling chromatin would just be to go up one step in your coarse graining and model the chromatin fibre directly. So we did that. So now we have a chain of spheres which represents the chromatin, and so now we use beads which have a diameter of about 30 nanometers, and so that's about 3 kilobase pairs of DNA. And the chromatin fibre has a persistence length somewhere in the range of 40 to 200 nanometers. So it's a more flexible fibre than the DNA if you think in terms of the ratio of the diameter to the persistence length. So in our simulations, the main difference is that this is a more flexible fibre. And so most proteins will be smaller than these 30 nanometer beads. Okay, so again we did simulations with a large DNA, lots of proteins, so a large chromatin fibre this time, and again we see this clustering due to the same effects I mentioned before. Okay, so just to summarize what I've talked about so far in this first part of the talk, so we've looked at a zoo of different very simple protein models, and we've looked at polymers with different flexibilities representing naked DNA and a chromatin fibre, and all of them have been shown to exhibit this clustering behavior. Okay, so when the DNA associates with the proteins, there's an energy reduction due to the protein-DNA interaction, but then there's also an energetic cost due to DNA deformation and also an entropic cost of bringing different DNA segments together. But by clustering the proteins together, we're able to reduce some of these costs. Okay, so I'll move on to the second part now, where we wanted to try and do some larger simulations and try and be a bit more realistic and have some links to some experimental data, although qualitative links at this stage. So we looked at having sequence-specific protein-DNA interactions. So we again consider a chromatin fibre. So these are 30 nanometer sized beads. And we looked at the interaction between human chromosomes 5, 8, 14, and 17, and two proteins, Pol2 polymerase and one of its transcription factors, P65. Okay, and so we identified chromatin beads which were sticky for the Pol2, and beads which were sticky for the P65, or both, and then beads which had only the hardcore steric repulsion for the proteins. Okay, so these proteins are often found in the bridges between different segments of the same chromosome and between chromosomes, and we use data from chip experiments to find positions on the chromosomes where these proteins commonly bind, and also 3C experiments give data about contacts between different parts of the chromosomes. This is data from Peter Cook's lab in Oxford. And so in our simulations we identified monomers and classified them either as binding or non-binding for each of the two proteins. And then we did some large simulations where we put 4 chromosomes in a simulation volume which was a cube of 3 microns in size, and we had 5,000 of each of the proteins, which had a diameter of around 10 nanometers. And then these were parallel simulations with about 200,000 monomers in total, and it took around 10 to the 8 simulation time steps, and that was on a 48 core machine that took about 2 weeks to run. So they're quite large scale simulations. And again, there's no attraction between the proteins and no attraction between the different chromatin monomers. Okay, and we see that we still get this clustering. So this top picture here shows the 4 different chromosomes, and so since we know that the chromosomes are found in territories, we initialize the system with 4 chromosomes separated in space and then equilibrated that, and we still see that they're in territories. And we still see these clusters. So this picture shows the system with the DNA made invisible and just the two proteins here. So you can see that these are in clusters. And these clusters contain both Pol2 and P65, although I think you can sort of see that, that often each individual protein sort of segregates. And this is just since each of these clusters has some of the chromatin fiber winding through it, and different parts of that fiber are sticky for the different proteins. Okay, and at these realistic kind of volume fractions, so about 30%, that you would expect that these crowding effects, such as the depletion interaction, would also be present, and that should favour clustering as well. Okay, so this sort of supports this idea of transcription factories. So you have these regions where you have the polymerase and the transcription factor localized in clusters, so that sort of supports this picture. Okay, so we can also plot a contact map from the chromatin. So this just shows which of the chromatin beads was in contact with all of the other chromatin beads, and by in contact we just mean that in the simulation they were within a certain volume. And these blue lines sort of separate the four different chromosomes which we looked at in the simulation, and you can see there are a lot of contacts between the or within each chromosome, and then there are also some contacts between the chromosomes. And our sort of goal is to try and compare this with chromatin contact data from 3C experiments. But we find that this contact map, these results depend very much on the initial conditions which we use in the simulation. So these are very long polymers, so they have a very long relaxation time, longer than the times which we've simulated so far. So we're also hoping to be able to run this on a larger computer and see if we can do this for a bit longer. But nevertheless, we can still make some comparisons. So if you look at all of the chromatin, then about 3% of the chromatin we've identified as having a propensity to bind with these two proteins. And if you look at the chromatin which is involved in the clusters, so the parts of the chromatin which are in contact, and you look at how many of those, how much of that chromatin likes to bind to these two proteins, then in our simulations we find about 20%, so it's enriched for the chromatin which is involved in the clusters. And this is also the case, although to a lesser extent, in real chromatin. Okay, so there's a qualitative kind of comparison there. Okay, so I'm getting towards the end now. So just to summarize, we found that there's a general propensity for proteins acting non-specifically with DNA to form clusters, even though there's no explicit attraction between them. And this clustering can reduce energetic and entropic costs of deforming the DNA, and perhaps this depletion interaction is also having an effect. Even if we make the protein-DNA interaction sequence-specific, clusters also form, and that's consistent with this idea of transcription factories. And finally, we can look at a contact map from these simulations and compare them with experimental data, and we want to do some more of that in the future. Okay, I've got a little bit of time left.
You've got about 5 minutes.
Okay, so I have just one more video, just because I thought it was quite a cool one, if I had time at the end. So this is a chromatin fiber this time, but it's just a small fragment of one of the chromosomes. So this is using the data where the pol 2 likes to stick to the chromatin, so we're only including one of the two proteins which I mentioned before. And it's just play that video. Okay, so the proteins here like to stick to some parts of this fiber. And you can see that clusters are starting to form, and I don't know how clear it is, but if you look closely, then you can see that around these clusters there are sort of rosette type features. So you can see these loops of DNA forming these rosettes, and these have been seen with chromatin fiber in vitro before. And I just thought it was quite cool how quickly that seems to emerge, just due to this this clustering effect. Okay. So clusters form accompanied by these chromatin loops, which look a bit like rosettes. And the finite size of the clusters, we think, is due to just the steric hindrance of the chromatin fiber. So you can only get so much chromatin in these clusters. Okay, and so then I'll now finish just by acknowledging again Davide Marandozo in Edinburgh and Peter Cook in Oxford and his team, who we've used their data for the simulations there. And thank you.
Thank you, Chris. That was ace. Lynn.
So I just have a comment. So now that you've built all these proteins, how about if you were to consider what new DNA is made as it would start being assembled into chromatin? And so first of all, the ends are never free like that. The ends are very special. They're always fixed.
Yeah.
So you've got your ends flopping around, and I don't know, it's not going to make a huge contribution. But how about if you were to spool DNA in slowly with the end fixed? I wonder if you would still see clustering, or would you start maybe better approximating closer to the...
Yeah.
It's just a suggestion. I don't know, because you never in the cell have the big ball of naked DNA and suddenly a big bunch of proteins, right?
Yeah.
It comes spooling in. Proteins may be there, but it's a different kind of...
Yeah, that would be interesting to do, yeah.
Chris, how difficult would it be to introduce attraction between protein monomers?
If you want short range attraction between the monomers, that would be easy to do. So if you want long range interactions, then it's going to slow the simulations down.
Short range would be.
Yeah.
Because you're already seeing clustering. This should give probably much more ordered clustering more rapidly.
Yeah.
Chris, two things. So, do you looked at the contact maps? Do you look at this, whether the distance graph, whether you get these fractal globule exponents or anything like
We
that?
haven't looked at that yet, no. But again, would be an interesting.
And the other thing is, do you ever see any knots trapped in those loops that get formed?
So again, we haven't really looked at it, but I would imagine that you will see that. Yeah.
So at the back, sorry, you're all fuzzy at the back, so yes, please.
So do I understand right that when you have specifically bind proteins, they can still bind several strands of DNA at the same time?
Yeah, so you have the DNA with just a string of beads, and some of those beads are sticky for the proteins. And in those last simulations, the proteins were just spheres, so two beads in the chromatin can make multiple contacts with the...
But that goes contrary to all we know about polymerases. A polymerase cannot bind two DNA strands at the same time. And the same about most of the transcription factors. Some proteins can bridge, but that's a very specific class of proteins.
So
most of the proteins, and polymerases cannot do this.
Yeah, so there are some proteins which can. So that's another thing we should think about. So for example, in the previous parts where we had the bridgers, which were like the HNS proteins, so they they dimerize, I think. So they each only have one binding site, but they dimerize. So perhaps there are other proteins which do that.
Right. CTCF can do this, for example, in eukaryotes, but polymerases are intrinsically incapable of doing it.
Yeah. So another thing which we're hoping to do is try and build a coarse-grained protein which really can only make one contact with the DNA, and then see if these effects are still working. So if they somehow bend the DNA in a way which, if some more of them were brought together, that would reduce the bending, and also this depletion interaction effect could drive the clustering there.
One more question for Chris. Yes, please.
Question is related with the chains are truly flexible, or?
So for the first half of the top, those had a persistence length of 50 nanometers. So that's about right for naked DNA. And then for the chromatin one, we had they're not completely flexible. They still have a persistent length, but it's much shorter compared to the diameter. So they're more flexible than in the first set of simulations.
There is any relation between the size of the particles and the persistence length and the size of the clustering that you are getting?
We haven't looked at that yet, so this is quite new work, and we're hoping to look at those things in more detail.
Thank you.
Brilliant. Well, should we thank both speakers again?